Beyond neurons, how support cells drive the feeling of fullness for decades, the scientific consensus on appetite was relatively straightforward. Neurons, the brain's primary signaling cells, were the undisputed masters of hunger and satiety. However, a groundbreaking study published in Proceedings of the National Academy of Sciences, PNAS, is shifting that paradigm. Researchers have discovered that the sensation of being full is not just a neuronal process, but a sophisticated relay involving cells previously dismissed as mere support staff. The hidden communication chain the research, a collaborative effort between the University of Maryland and the University of Concepcion in Chile, focuses on the hypothalamus, the brain's command center for metabolic regulation. The study identifies a complex, multi-step communication circuit that bridges the gap between eating and feeling satisfied. The process follows a specific biological relay. Detection. Specialized cells called tanocytes detect glucose levels after a meal. Conversion. Instead of just signaling the brain, these tanocytes process the sugar and release a byproduct called lactate. Transmission. This lactate travels to neighboring astrocyte cells long thought to exist only to support neurons. Activation. Astrocytes possess specific receptors, HCAR1, that sense the lactate. Once activated, these astrocytes release glutamate, a chemical signal that tells appetite-suppressing neurons to fire. A dual-action mechanism. One of the most striking findings of the study is how this lactate-driven circuit manages the brain's hunger thermostat. The hypothalamus contains two opposing forces: neurons that drive hunger and neurons that suppress it. The researchers discovered that lactate may act as a biological double brake on appetite. Indirectly, it triggers the fullness neurons via the astrocyte glutamate pathway. Directly, it appears to simultaneously quiet the hunger-promoting neurons through a separate route. This dual effect suggests that the brain doesn't just signal that it is full. It actively works to shut down the urge to eat from two different directions at once. Why this matters for future medicine historically? Astrocytes were viewed as the glue of the brain, essential for structure and maintenance, but not for decision-making or behavior. This study overturns that assumption, proving that these cells are active participants in how we experience physiological urges. The implications for clinical treatment are significant. Targeted therapies. If scientists can learn to manipulate the HCAR1 receptor on astrocytes, they might be able to induce satiety without the side effects often associated with traditional neurological drugs. New approaches to obesity. Current treatments for obesity often target neurons directly. This research suggests that targeting the support cells could offer a more nuanced and effective way to manage appetite and eating disorders. While these findings are currently based on animal models, the presence of tanocytes and astrocytes in all mammals, including humans, makes this a highly promising avenue for medical advancement. Conclusion: by uncovering the critical role of astrocytes and tanocytes, this research reveals that satiety is a much more integrated, multicellular process than previously understood. This discovery opens a new frontier in metabolic science, potentially providing the blueprint for next-generation treatments for obesity and appetite regulation.